Now this is the fourth part of lecture 2, and in this part I'll talk about fermentation media. Okay, so what is fermentation media or food media? We all know media is the source of nutrients of the microbes. Okay, so the microbes, for the every organism needs food, nutrients for their survivability, for their growth. Okay, so microbes are not any exceptional things. They also need, when they grow, they also need different kind of nutrients. Okay, so and they get this from the media. Okay, so if we want to culture our microbes, we need to establish the optimum composition of the media. Okay, that means we have to find out the optimum composition of the media which will most favorable for the growth of that microbe. Okay, and usually it is done in the lab. Okay, in the lab scale, when we isolate and do further uh, development. Okay, in that time basically we need to optimize the composition, and how we can do that? We just can try different substances, right? We can use add water, and we can try different carbon source, nitrogen source, minerals, and other supplements, and we can add them in different concentration. Okay, and by doing so we have to finalize or establish the optimum media composition. Okay, however, I mean, for industry level, sometimes the media composition that is most optimum for growth in the lab may not be optimum for the growth in industrial level, because in the industrial level we grow them. In the fermenter, we grow them in very large scale. The condition is very different there from the lab. Okay, in the lab we culture them in a small amount, like a few uh, milliliter or maybe 1 or 2 liter. Okay, but mostly 5 liters. Okay, but in industry level we culture them in thousands liter. Okay, so the pressure is different in the fermenter, the availability of the nutrients is different, oxygen availability is different. Okay, so that's why the media composition that we use, that we establish in the lab scale, may not be optimum for the industry scale. Okay, anyway, the media used in the for industrial fermentation should have some criteria. Okay, and these are: it should give maximum yield of production, it, it should give minimum yield of undesired product, it should be consistently available throughout the year. Okay, and it should be cheap. It will cause minimum problem in other aspects of production and agitation, extraction, purification, and waste treatment. Okay, these 2 points are actually important for the industry, like which are the materials that you used as a part of the media should be available throughout the year. Okay, so suppose the raw material you use to prepare the media is available during from like in the winter. So what you will do in the summer? Okay, if you don't have the raw materials, you cannot run your fermentation. Okay, so it's better if we can choose the raw material which is available throughout the year, and also it should be cheap. Okay, we cannot use costly raw material, right? If we have to use costly raw material, our final product will be very costly. So we should always try to select a cheap raw materials. Okay, these were the criteria of the media that are used in industrial fermentation. Now, if we think about the media composition that is used in the industry level, okay, in that case we should use cheap materials such as, for example, cane molasses, corn strip liquor, sugar beet juice. This kind of cheap or inexpensive sources are used as a media component. Okay, however, for some sensitive fermentation, okay, pure form of glucose, sucrose, and other carbohydrates are also used. Okay, but these are expensive. Okay, so we will use this only when the fermentation process is sensitive. Okay, and sometimes for some specific products, we need to add some extra materials, such as for production of amylase we have to add starch. Okay, and similarly, if we want to produce proteins, nucleic acids, and other essential cellular components, in that case, we have to add some materials, raw materials in the media, which will, and from which the microorganisms can produce this one. I mean, the microorganisms will use those raw materials and produce proteins, nucleic acids. So, in lab scale, if we, I mean, in laboratory, if you want to produce proteins, nucleic acid, etc., in that case, we, I mean, as media components, we add partially digested peptone or tryptone or beef extract. Okay, and the microbes they utilize this and produce proteins, nucleic acid, etc. However, these are costly. I mean, digested peptone, tryptone, beef extract, these are costly. So, if we want to produce proteins, nucleic acid at industry level, we cannot use this as raw materials. Instead, we have to provide a cheaper alternative, such as soy meal or ammonia or nitrate salts as a as the nitrogen source. Okay, and sorry, and also the media they contains growth factors, vitamins, anti-foaming agents, uh, precursors, inducers, chelating agents, stress elements, etc. Okay, so that means the media, okay, doesn't mean it's a media used in the laboratory or in the industry level. It should contain different kind of raw materials. Okay, so some raw materials will be act as a carbon source, nitrogen source, or vitamin source, etc. Okay. So in the from the next slide, we will learn about those. First, carbon source. Now, carbon source. So carbon is required for all the biosynthesis process, and also it serves as the energy source. And in industry, we can use different raw materials to provide the uh, carbon source. That means different nutrient requirements, such as sugar cane, sugar beet molasses for as a source of sucrose, corn sugar, starch, cellulose as a source of glucose, milk whey as a source of lactose, vegetable oil as a source of fats, etc. Okay, so now which raw materials we'll use as carbon source? It is influenced by different factors, such as cost of the product and cost of the raw materials, cellular yield coefficient, rate at which it is metabolized, government regulations, and geographical locations. These two are very important factors, that is, geographical locations and governmental regulations, such as like in Bangladesh, you want to establish a fermentation industry. Okay, so you cannot use everything as raw materials. Like for example, you want to produce alcohol. So grape is a very good raw material for the production of alcohol. But can you use grape in Bangladesh? Definitely you can. In that case, you have to import it. Okay, but if you have to import your raw materials, maybe the cost will be so high that you cannot set a competitive price for of your product. Okay, and also like, so that's why I mean in Bangladesh, okay, there is only one company, Kedu and Kedu, who produce alcohol, and they use molasses. Okay, in Bangladesh, uh, rice is also available, right? Rice, I mean, we cultivate lots of rice, potatoes, wheats. So this can also be used to produce alcohol, but why we don't use that? We have a very large population, right? We just cannot use rice to produce alcohol. Maybe we can. I mean, theoretically we can, but our government regulations will not allow us to use rice to produce alcohol. Okay, so both geographical locations and government regulations are important. Based on our geography, we should choose the raw material. I mean, which raw material is available in that geographical place. Okay, and also government regulations, because government regulations prevent some raw materials to be used as a carbon source for the fermenter. Okay, so I will give you one example. Say, in Japan, they use rice to produce high quality uh, alcohol. Okay, but they cannot use, but they actually have to use the Japanese rice to do that. But Japanese rice is kind of costly. I mean, rather if they import rice from other country, it will be cheaper for the industry. But the government regulations is such that they must first they have to use all the rice. I mean, they have to use Japanese rice. Then, if required or necessary, they can import the rice. Okay, so government regulations can influence greatly. I mean, greatly your decision which raw materials you will use. Okay. Then nitrogen source. So most industrial microbes can utilize both inorganic and organic nitrogen sources. Okay, and inorganic nitrogen may be supplied as ammonium salts, ammonium sulfate, and diammonium hydrogen phosphate or ammonia. Okay. In addition to serve as a nitrogen source, ammonia can also be used to adjust pH of the fermentation. I mean, if we use ammonia, it will serve two purpose, right? It will serve as a nitrogen source. Also, it will help to adjust the pH. Okay. And in case of organic nitrogen, the organic nitrogen sources include amino acids, proteins, and urea. Okay, these are some example of organic nitrogen source. Corn, yeast liquor, yeast extracts, peptones, soybean meal, etc. Okay. Microorganisms also require certain mineral elements for their growth and metabolism, so that's why you also have to add different minerals. For example, in many media, magnesium, phosphorus, sulfur, calcium, chlorine, etc. must be added, and I mean this must be added in many media. Also, in some media, some other minerals such as cobalt, iron need to be added. Okay, and also in some media we have to add chelators because in some media we use metal phosphates, okay, as raw materials or the media components, but metal phosphates they tend to precipitate. Okay, so it's a problem, right? If they just form precipitation, then it will be difficult for the microbes to use that phosphate. So we need to use chelating agents such as EDTA, citric acid, etc. Okay, so what these chelators will do, they will form complex with this the metal ions. Then uh, the microorganisms they will be able to gradually utilize the those metals, okay, those chelated metals. Okay, however, it is important to check the concentration of chelators because some chelators they may interfere with the growth, they may inhibit the growth of the bacteria. For example, EDTA. Okay, if you use too much EDTA can affect the growth, they can inhibit the growth of the cells. Okay, so you should check that what concentration I mean at what concentration you will use the chelators. Okay, and in many media, okay, during media we use the chelators separately after autoclaving because sometimes I mean after autoclaving the chelators can be destroyed or denatured by the autoclaving. So that's why we need to, I mean, first we'll prepare the media, we'll autoclave them, then we'll add the heat sensitive chelators. Okay, and also we have to, sometimes we may need to add vitamins and growth factors. Usually many bacteria they synthesize all necessary vitamins from basic elements. However, for some microorganisms, such as there are some bacteria, filamentous fungi, yeasts, and yeasts, okay, they cannot produce all the vitamins. So we may need to add vitamins, okay, as supplement for the fermentation. Okay, and also, usually most natural and carbon sources that are used in the fermentation media contain some required vitamins as minor contaminants. Okay. Sometimes for some fermentation process, we also need to add precursors. Okay, precursors are those substances. which will which the microorganisms will utilize to produce the product. Such as, for example, if you want to produce penicillin G using Penicillium chrysogenum. Okay, so if you want to use Penicillium chrysogenum, this microbe, to produce penicillin G, you have to add this precursor there. Okay, phenyl acetic acid. Okay, because Penicillium chrysogenum, they utilize this precursor to produce penicillin G. Okay. Anyway, precursor can be defined as substances added prior to or simultaneously with the fermentation which are incorporated without any major change into the molecule of the fermentation product, and which generally serve to increase the yield or improve the quality of the product. In some fermentation media, we also need to add inducers. Okay, inducers are some substances which induce our microbes to produce the product. For example, many production of industrial enzymes can be induced by adding some inducers. Okay, so and also actually for some product we must have to uh, add the inducers, because production of those products are dependent on inducers. Okay, and also inhibitors are added in some fermentation media because they inhibitors, they can serve different purposes. For example, they are used to redirect metabolism towards the target product and reduced formation of other metabolic intermediates. Otherwise, halt a pathway at a certain point to prevent further metabolism of the target product. Okay, so for example, like if during the production of glycerol using Saccharomyces cerevisiae, sodium bisulfite is used. Okay. Sodium bisulfite can increase the production of glycerol, because sodium bisulfite, I mean the Saccharomyces cerevisiae, they produce glycerol, and also they produce acetaldehyde. Okay, during their growth. So if we add sodium bisulfite, this sodium bisulfite inhibit the acetaldehyde production. Okay, so this yeast cannot produce the acetaldehyde, so they produce more glycerol. Okay, and another example is if you I mean if you want to like for the production of glutamic acid using Micrococcus glutamicus. Okay, penicillin is added. What penicillin do? Penicillin inhibit cell wall formation. So if we add penicillin in this fermentation media, they will increase the cell wall permeability. Okay, so the Micrococcus glutamicus, they will produce the glutamic acid inside their cell, and they will easily release this into the media. Okay. Okay, and then water. Okay, I think I don't need to explain why water is important. Right? Water is the most important part of the media. Okay, so usually in all the fermentation process except SSF, single cell, okay, except the SSF production, lots of water is necessary. Okay, however, uh, not only for the fermentation process itself. Okay, water is also used for various purposes in the fermenter. For example, like for heating, cooling, cleaning, rinsing, etc. Okay, and so where you will establish your fermenter industry, you should ensure that you have access for clean and large amount of water. Okay, and the water should be in the quality of that water should be consistent, because if the water is if the quality of the water is not consistent, it will affect your fermentation. Okay, it always do that. So composition of the water is actually very important to produce a consistent quality product. Okay, and however, anyway, in the industry, we should try to reuse the water, because I mean, water is not free. Okay, I mean, if you have to use large quantity of water, and if you don't use them, your cost will greatly increase. Okay, so if you can reuse the water, you just can reduce the water cost by almost 50%. Okay, and not only that, if you cannot reuse the water, that means you have to treat lots of influent. Okay, it will add some extra cost, and that extra cost is not small. Okay, it can increase your effluent treatment cost almost by tenfold. Okay, so if you can reuse the water, that means you have to treat less effluent, thus save some money. Then oxygen. Okay, and okay, oxygen is necessary, right? Many organisms, they require oxygen for actually oxygen for the actually, except the anaerobic microbes. All the microbes, they need oxygen for their growth. So we have to supply sufficient oxygen into the media. Okay, so unlike the and the oxygen supply should be continuous. Okay, and we supply oxygen in the fermenter media by using usually asperger, and you already know about the asperger. Okay, now how much oxygen we have to add into the media, it depends on the microorganism. Some microorganisms, they need a high amount of oxygen. In that case, we may have to supply pure oxygen in the fermenter. Okay, some microbes, they may require less amount of oxygen. In that case, we will not supply pure oxygen, rather we will mix oxygen with the air, and then we will supply this mixture into the fermenter media. Okay, and the organisms, I mean the oxygen requirements by the organism, it's vary greatly depending on what type of carbon source they are using. Okay, and for most fermentations, we need to sterile the air or oxygen before we put them into the fermenter. Okay, and for industry, uh, filter sterilization, I mean filter sterilization is most famous for sterilizing air or oxygen. We also have to add antifoams in many fermentation process, because in many fermentation process, lots of foams are produced, and foams are problematic things. Okay, because they will occupy the headspace of the fermenter. Also, they will interfere with the gas disengagement. Okay, so during fermentation, okay, many toxic gas can be produced, right? So this gas need to be, I mean, get out from the media, and this occurs through the gas disengagement. And this gas disengagement, it happens in the head space. I mean, in the head space, there is air and the media. Okay, in this area, uh, this junction, the gas disengagement occurs. But if the foam is there, so they will interfere with the gas disengagement. So ultimately, they will interfere with the fermentation process. Right. So it's important to remove the foam, and to remove foams, I mean, foams can be removed by mechanically, but sometimes the mechanical process, I mean, is not adequate enough. Okay, or in every fermenter, we cannot just use mechanical foam remover. So sometimes chemical agents are used, which we call antifoams. Okay, these antifoams are necessary to reduce foam formation during the fermentation. And foam are largely formed by the media proteins. Okay, the proteins which are present in the media. So once these proteins, they reach in the air media interface, they get denatured and form form a stable skin-like foam layer. Okay. Anyway, the an ideal anti-foam that is used in the fermenter should have these following properties: disperse readily and have fast action, active at low concentration, because if we have to use high concentration of antifoam, they may interfere with the growth of the microorganisms, that means with the fermentation process. Long acting in preventing new foam, should, I mean, the life of the antifoam should be long. Okay, should not be metabolized. I mean, if they get metabolized by the by the fermentation organism, okay, they cannot do their work, right? So they should not be get metabolized by the microbes, should not be toxic to microorganism, humans, etc. Okay, and they should be cheap, okay, and should not cause problem in the fermentation. Okay, so that was all for lecture 2. Thank you for your patience.